Right, hi everyone. Welcome to the second study unit. We're going to have to go through neurotransmitters again, structure of the neuron, and then look at various ways that the brain is scanned. That's the gist of what we're covering, and also just parts of the brain. So let's get through that. Right. So let's start with the neuron. The neuron is the nerve cell, and there are millions and millions and millions of them inside the brain. What you've got is you've got the soma or cell body. Inside is the cell nucleus, which is where the DNA is. So the one side of the soma, you've got the axon. That is the receiving part of the neuron. Okay, and you get a long, sometimes you get a long axon, which is coated in myelin, a fatty insulation. If the myelin sheath is there, it has nodes of Ranvier in which there is no myelin. So that it's little sausages of myelin around the nerve, and where it's pinched off, that's the node of Ranvier. So axon coming into the soma. On the other side, you get the dendrites, which look like roots. At the end of each little root, there's a terminal button, a little blob, okay? And that is the end of the dendrite. From the terminal button, neurotransmitters get released and they cross the synaptic gap, the gap between the one neuron's dendrites and the next neuron's axon is called a synapse. And through there passes neurotransmitters. Okay, so that's the structure of the neuron, which we do need to know. And the next thing we need to look at is the neurotransmitters. So across that synaptic gap, neurotransmitters get sent. So you get the presynaptic membrane, which is the dendrite side, the terminal buttons, and the postsynaptic membrane, which is on the axon. You get three kinds of neurotransmitters: monoamine, which includes dopamine, serotonin, and acetylcholine. These are based on monoamino acids. Okay, so choline, tryptophan, and what is dopamine based on? Tyrosine. Okay, so that's the one kind, monoamine. Second type is the simple amino acid neurotransmitter, such as GABA, which is both an amino acid and a neurotransmitter. Then thirdly, you get neuropeptides. Peptides are little chains of proteins, okay? Because each amino acid is made of a very particular molecular structure. These are not connected to exact amino acid, they're just protein chains. For example, epinephrine and endorphins. Endorphins are examples of neuropeptides. Okay, epinephrine, sorry, those are monoamines. So the epinephrine is the same word meaning as adrenaline. Norepinephrine, same meaning as noradrenaline, and those are monoamines. Okay, so let's look at each of those neurotransmitters and just remind ourselves of the functions. So if it comes up in the questions, we're ready for them. Okay, so we've got acetylcholine. This is, a lot of it's found in the hippocampus. There's a lot of acetylcholine in the hippocampus, so we know that it's associated with memory. Dopamine. Okay, not enough dopamine is associated with Parkinson's disease and Parkinsonian symptoms. Okay, like such as tremors and limb rigidity. Too much dopamine is associated with some forms of schizophrenia. Epinephrine and norepinephrine have to do with fight and flight and also with anger. Okay, serotonin has to do with regulating, inhibiting dreaming. Deficiency is associated with severe depression. Okay. GABA is associated with some learning and memory. Glutamine is also associated with learning and memory. And an example of a neuropeptide, endorphin, regulates pain and also brings pleasure. Like if you run, you get a rush of endorphins, which is very pleasurable. Okay. Dopamine is also associated with motivation, rewards, and reinforcement. So it's closely associated with addictive behavior, such as gambling or scrolling on your phone. Okay. Right, let's have a quick look at the structure. The nervous system is divided into two parts. The central nervous system is the brain and the spinal cord. The peripheral, you can then subdivide into the somatic, which is the sensory coming in, and the motor going out, telling your body what to do. That's conscious. Then you get the autonomic. The autonomic nervous system is the second part of the peripheral nervous system. And you can divide that into the sympathetic and the parasympathetic. The sympathetic gets you going and makes you act and respond. The parasympathetic calms you down. It slows you down. So it's for resting and recovery and digestion and so on. Okay, so those two together balance the body's activity and rest, excitation and inhibition. Okay, so the next thing we're looking at is the question of how do we understand the brain? So the most primitive and obvious way is to cut brains open and look at them post-mortem. And this does give interesting information. You can see tumors, you can see damage to the brain, such as the very particular damage that's associated with Alzheimer's, where you get all these conglomerates of tissue, damaged tissue in the brain. But we don't know what's the cause and what's the effect. So causality you cannot really see by dissecting brains. There's animal studies, which are often very creepy, and you can get more information because you can do horrible experiments on poor animals that happen to be alive at the time. And then you can cut them open afterwards as well. Okay, then more interesting and more innovative and less invasive are ways of recording brain activities. So the first of these is the electrical recordings, the EEGs. EEG stands for electroencephalogram. And with an electroencephalogram, you can measure brain activity over time because you can measure brain waves, literally measuring brain waves. And so you can see which parts of the brain are busy when. But EEGs only work in periods of several seconds. They're quite slow, and brain activity is in milliseconds. So that's not optimum. By taking a series of them, you can get a sense of the flow of brain activity over time. This is called event-related potentials, ERPs, which is a series of EEGs. Okay, then let's look at other methods. Static imaging techniques. Okay, that's magnetic resonance imaging. It's almost like taking a photograph of the brain in time. X-ray techniques such as angiogram and CT scan. Okay. So you put a magnetic field through the brain and you rotate scans and you get these like 3D slices of the brain that you can then look at. And this is useful for seeing things like lesions and tumors. Metabolic imaging. Okay, so by looking at how much oxygen or how much glucose the brain is using, you can see which part of the brain is active, because wherever it's active, it's going to be using a lot of oxygen and a lot of glucose. The brain is a very greedy organ. It uses up a massive amount of our metabolic energy. So let's see. We've got how do you measure? So which one works with glucose? The one that works with glucose is the PET, positron emission tomography. So what you do is you give people a form of glucose that's mildly radioactive. So it's giving off radiation. And then you use the positron emissions from that radiation, and you create a map of it, a tomography, to show you which parts of the brain are burning up glucose. So you can see which parts of the brain are working. Okay. So you can see, oh, yeah, look, this person is looking at something and it lights up the visual cortex in the brain. You can see that visual cortex working. The more you think, the more glucose you use. And people use their brains very differently depending on their measured intelligence, which is interesting. Okay, so now if we want to look at how to measure the brain activity looking at oxygen, that's where we would use functional magnetic resonance imaging, fMRIs, okay? So use the MRI, but you're measuring the oxygen use. How do you do that? Well, molecules of oxygen respond differently to a magnetic field. So what you do is you hit the brain with a magnetic field, and the skull is porous to magnetic fields. And you can literally see where the oxygen is being used because it lights up those particular areas of the brain. So you can see what is happening. That's a very nice high temporal resolution. So you can see what's happening in fractions per second. So fMRIs and PETs are very, very good for getting ideas of what the brain is doing and where. Okay, so let's look at the different parts of the brain. We can divide it into the forebrain, the midbrain, and the hindbrain. The hindbrain sits at the top of the spinal cord. Okay, so the forebrain is where most of the higher functioning, most of what makes you happens. So the forebrain has the very important cerebral cortex, which is where most of your thinking, receiving sensory information, cognitive processing, planning, deciding how to move your body happens, okay? The limbic system is the second part of the forebrain after the cerebral cortex. The limbic system consists of the hippocampus, the amygdala, and the septum. Hippocampus is associated with memory, amygdala with fear and identifying danger, and the septum influences anger and fear, okay? Amygdala is associated with anger and aggression as well. All right. Third part of the forebrain is the thalamus. The thalamus is like the relay station for sensory information coming into the brain and directing it to the different parts where it is processed within the brain. Okay. Then the hypothalamus is the endocrine part of the brain and manages the autonomic nervous system, neurotransmitters, hormones, temperature regulation, and basic behavior like eating and mating, fighting and fleeing. Okay. All right, so moving on to the midbrain. The midbrain, we're looking at, okay, there's a few different regions. We're going to talk about the superior colliculi and the inferior colliculi. Superior colliculi, meaning above, superior, is involved in vision, and the inferior colliculi is involved in hearing. So there's like this, how to make the sensory organs connect to the brain. That happens in the midbrain or in the thalamus, okay? You also get the reticular articulating system. So this is also important in controlling consciousness, sleep and arousal, attention, cardiorespiratory function, and movement. So keeping the body ticking along. Now the other part of the brain is very, very important for those autonomic mechanical functioning of being alive is the hindbrain. The hindbrain is also a relay station between the spinal cord and the brain. You've got the cerebellum, meaning little brain, which is where your balance and coordination comes from. If you've got cerebral palsy, that's the part of brain that's damaged, and that gives you that inability to manage your motor controls or to inhibit motor movement. Okay. The pons, which is again involved in sleep and arousal, and the medulla oblongata, which is involved in cardiorespiratory function, digestion, and swallowing. So there's been people whose brains have been totally functional and they've gone into a coma because they've got a tumor in the hindbrain. You know, and sometimes people were like, I think I read about this one surgeon, this person was an organ donor. And he realized there was a tumor in the hindbrain. They operated on the tumor. that person woke from the coma and was completely functional, but until they removed that tumor from the hindbrain, they were in a vegetative state. They were unable to move, unable to be conscious or communicate. They were just comatose. So the hindbrain is very, very, very important for keeping us alive. It's evolutionarily the oldest part of the brain, and it's the first part to develop in the fetus, in the developing fetus. Okay. We're not going to go into more detail. Hopefully that will cover us. Okay, so there's been fascinating experiments with the left and right hemisphere of the brain, particularly with people who've had brain damage or unfortunately had to have surgery to save their lives on the brain that's restricted the brain functioning. So what they do is when they find people like that, they become research subjects for numerous experiments where you identify, okay, this person cognitively cannot see on this side of their visual field, and that would be associated with the visual cortex on the opposite side. But they can see on this side, and you do all sorts of interesting experiments where you ask the person what they see. Are you seeing an orange or an apple? And the person will answer an orange, but they'll draw an apple or something like that. So it's very, very interesting, very, very interesting, rather unsettling experiments can be done with people with brain damage to discover how the brain works for all of us. Okay, let's quickly run through the 4 lobes of the cerebral cortex. You've got the frontal lobe, which is involved with the higher brain functions, decisions, planning, and self-inhibition. Okay, so Phineas Gage was a railway worker. He got a railway spike through his frontal lobe, the left frontal lobe. And although his intellect was not affected, he became reckless, he became rude, he started drinking to excess, and died penniless because no one could employ him because he was now unreliable and impulsive, whereas before he'd been a very steady human being with a functioning frontal lobe. Okay, then you get the parietal lobes on the sides. Okay, and they manage somatosensory processing. What sensations do you have in the skin and the muscles? You get the temporal lobes, which have to do with hearing and which are actually just above and behind the ears. And then you get the occipital lobe, which is like the opposite side of your head to your eyes, and which manage visual processing. Okay, and it crosses over, which is interesting. So this eye is managed by this lobe of the occipital lobe and vice versa. Okay. Now, what they did spend a lot of time looking at when they were studying the brain and learning how it was working was association areas. Where you look at, oh, okay, this part of the brain is associated with this function, like processing images in this particular way, okay? And they mapped out different association areas in the brain with partial success. And that wraps up chapter 2. We're ready to go on to chapter 3. I hope that was helpful, and good luck. See you in the next video.